Hi future scientists, I am Atom. Today we will dive into independent and dependent variables, formulate a scientific hypothesis, understand controls, and learn how to graph data in an experiment. Problem statement. How do different concentrations of hydrochloric acid affect its reaction with calcium carbonate in terms of the amount of carbon dioxide gas produced? Independent variable is the part of the experiment that we manipulate on purpose. It is what we as the experimenters decide to test. In our example, the independent variable is the concentration of hydrochloric acid. So the different hydrochloric acid concentrations, let's say, are 0.5 molar, 1 molar, 1.5 molar, and 2 molar. If you're wondering what this uppercase M is or what does molar mean, this is all you need to know for this lesson. Molarity is a measure of concentration in chemistry, and the uppercase M stands for molar, which is the unit used to express molarity. A higher molar value means higher concentration, and a lower molar value means lower concentration. We will learn about molarity in detail in a future lesson. The dependent variable is the effect or outcome that depends on the independent variable. Here, our dependent variable is the amount of carbon dioxide gas produced in a specific amount of time. To measure the volume of carbon dioxide produced within a certain time frame for each concentration, we can use the water displacement method. So what is the water displacement method? Here's how it works. Fill a graduated cylinder with water and invert it in a water bath. The reaction between the hydrochloric acid and calcium carbonate takes place in a flask connected to the graduated cylinder by a delivery tube. As CO2 is produced, it travels through the tube and displaces the water in the graduated cylinder. The volume of gas collected can then be read directly from the markings on the cylinder. Scientific hypothesis. Here's our scientific hypothesis in the if independent variable, then dependent variable because scientific reasoning format. If the concentration of hydrochloric acid increases, then the volume of carbon dioxide produced will increase because more acid molecules will be available to react with the calcium carbonate. Controls. Controls are the things we keep the same for every trial, so that the only thing affecting the outcome is our independent variable. For example, we'll keep the temperature, the mass of calcium carbonate, the time duration of CO2 collection, and the volume of hydrochloric acid constant for each trial. Why do we only have one independent variable in an experiment? If we change both the concentration of hydrochloric acid and the temperature of the solution, we would not know exactly what affected the amount of CO2 produced, whether it was because of the acid concentration, the temperature, or a combination of both. That is why we only have one independent variable in an experiment. Multiple trials ensure the reliability and accuracy of results by averaging out inconsistencies and reducing the impact of outliers and errors. Here is a fictitious sample data table with 5 different trials and the calculated averages for each concentration of hydrochloric acid. Graphing. We can plot the sample data on a graph, with each data point representing the average volume of CO2 produced for each concentration of HCl. The x-axis represents the independent variable with correct units. The concentration of HCl parenthesis M for molar. The y-axis represents a dependent variable with correct units. The amount of CO2 produced parenthesis ml for milliliter. The title of the graph should describe the relationship by stating the dependent variable versus the independent variable. So we could title this graph the volume of CO2 produced versus hydrochloric acid concentration. Now plot the data points. 0, 0 is a data point since 0 molar HCl means 0 ml of CO2 produced. Draw the best fit line. The graph shows that as the concentration of HCl increases, the average volume of CO2 produced also increases. There you have it. Variables, scientific hypothesis, graphing. Now you're ready to design your own experiments, graph your data, and discover the amazing ways variables interact. Please subscribe, like, and share for more fun science. Happy learning. Thank you.